So in today's video, I'll be talking about the basics of DNA and a little bit about RNA as well. Its primary structure, models explaining the structure, its types, stability of DNA and its quantification. So let's get right into it. In my previous lecture, I had already shared the details about the birth of genetics and the discoveries that led to the conclusion that DNA is the genetic material. So further, we'll be studying about those models, those scientific discoveries that have come up so as to describe the structure and features of DNA. But first things first, nature of the chemical subunits in DNA and RNA. Because if we don't know this, then we won't be able to understand the structure of DNA. So DNA is basically made up of three parts, that is phosphate group, ribose sugar with with, uh, with a hydrogen, that is no hydroxy group on the second carbon of the sugar. That is why it is called as deoxyribose and deoxyribonucleic acid. While in RNA, the hydroxyl group is present in the at the second carbon of the ribose sugar. So this is what it makes different from RNA. RNA is a single strand which consists of ribose, while DNA is a double strand helix which consists of deoxyribose. Another difference is in the base pairs, that is the DNA consists of cytosine, thymine, adenine and purine, while RNA does not consist of thymine, instead it consists of uracil. Alright. And it is the sugar and this phosphate, it is this sugar and this phosphate that forms the backbone of the double helical structure and even the RNA as well. It is the base which projects to inwards, and DNA is also called as the polynucleotide chain. So what is nucleotide? Nucleotide is sugar plus base plus phosphate. So uh, this entire forms a nucleotide, while this entire forms a nucleoside. So DNA is made up of polynucleotides. Alright. And the base pairs, that is adenine bonds with thymine using two hydrogen bonds, while guanine bonds with cytosine by three hydrogen bonds. Yes. So here is the molecular model stylized diagram and the chemical structure of DNA. You may see the chemical structure, and I'll be discussing this chemical structure in detail. Yes. So coming to the salient features of double helix of DNA is that it is a two polynucleotide chain and backbone is made up by sugar phosphate moiety. Sugar phosphate moiety background is there, you see, and the base project towards inward. And as I already had already discussed about the hydrogen bonding between the base pairs, and these two chains are coiled in right-handed fashion. Right-handed fashion means it goes like this. Okay, while left-handed fashion goes like this. So in this fashion, it is coiled. Right-handed fashion, it is coiled. And the pitch of the helix is 3.4 nanometer. Now what is meant by a pitch? So let me draw a DNA over here. See, this is your DNA. Okay, so the pitch is the distance from here, this groove, till this groove. So this is 3.4 nanometers and there are 10 base pairs in each turn. And the distance here, this is base pair, and the distance between base pairs in helix is 0.34 nanometers. See, you can automatically calculate the number of base pairs in each turn, that each pitch, this pitch has a distance of 3.4 nanometer, while the distance between each base pair is 0.34 nanometer. So it can be calculated up to 10. And it is due to the base pair stacking manner in this way that it forms hydrogen bonds vertically as well as horizontally. So it provides an utmost stability, and this provides the most stable structure to DNA, and it is this complementary base pairing that gives DNA a uniform diameter throughout the structure. I hope it's clear to you all. Okay, so now let's discuss in detail about the properties. Properties will be concerning more about the stability. Is that the distance between the two sugar phosphate backbones is always the same. It is always the same. I'll here draw DNA for your ease so that you can easily understand it. Yes. It is this base pair that it gives a uniform and regular shape to the DNA entirely, and DNA base pairs, this base pair I have here drawn it like that, but the but in actual structure the base pairs project perpendicularly to the sugar phosphate backbone. This sugar phosphate backbone, as you may know, that sugar and phosphate, they form a hydrophilic structure, but the bases inside, they are hydrophobic. And it's not only the interaction which occurs between the base pairs by 3 or 2 hydrogen bonds, as I told in the case of adenine and thymine, or guanine or cytosine, but there exists a perpendicular stacking hydrogen bonding which occurs there, and it is this bonding as well as this bonding which provides it a hydrophobic nature inside of the DNA, and it also conforms towards its stability, and this bonding also causes the sugar phosphate backbone to twist. And it is the spiral structure that places the base pair inside the helix and keeps the sugar phosphate towards the hydrophilic side, hydrophilic as in outwards, just to ensure that DNA base pairs are protected. Alright. Okay, so now we'll be discussing one by one the contribution of each scientist, that is Watson and Crick, Rosalind Franklin and Chargaff's rules. And a few points to mention here, that DNA is double stranded helix connected with with hydrogen bonds, and this hydrogen bonding rule and the base pairing is very well explained by Chargaff's rules. Most of the DNA is right handed coiling except for ZDNA, which has a left handed coiling. The DNA double helix is anti-parallel and it runs always it will run from 5 prime to 3 prime, and outer edges of the bases are available for potential hydrogen bonding. This outer edge, you can see outer edge is available for hydrogen bonding, and that is why it is forming hydrogen bonds. Alright. Okay, the very first point we'll be discussing is the X-ray diffraction studies given by Rosalind Franklin, which actually proved that DNA is a double helical structure, and most of its details I have discussed in my previous video. So in order to get the clarity about it, you may check out my previous video, which is entirely explaining about the X-ray diffraction studies and its interpretation. Yes, next coming to the Chargaff's rules. Chargaff's rules are two rules. With the second rule is a further explanation of the first rule. The amount of adenine equals to thymine and cytosine equals to guanine, which is very much obvious. We can get it from the structure. And obviously, if A is equals to T, then and C is equals to G, then the ratio will automatically be equal, and hence in a DNA we can calculate the number of base pairs, the percentage of base pairs, just by knowing one quantity of just by knowing the percentage of one base. And what is the significance of Chargaff rule? Chargaff rule provides the insight into the structure and behavior of DNA, and it provides specific base pairing rules forming the double helix, and it also understanding the DNA replication. And it is this base pairing which provides it a symmetrical structure as well as stability. See, it is a thumb rule of nature that whatever is which is symmetric, it will definitely be stable. So if these two are in if these base pairing is in symmetry, then the nature of DNA will automatically be stable and it will be in symmetric manner. Got it? And principles of complementary base pairing I have already discussed, and it provides stability also. So I won't be discussing it into detail here. And the equal proportions of complementary base pairs in DNA molecules also I have discussed. So I won't be going in much in detail about it. Yeah, but these Chargaff's rules help in understanding the DNA's structure and functioning. Also, it lays the foundation to comprehend the stable structure. That, see, base pairing is equals to stability, and the mechanism of this base pairing is critical for genetic information storage and retrieval. And also it is these Chargaff's rules that has paved way to the Watson and Crick's model of DNA, which I'll be telling further. Yes, Watson and Crick's model of DNA. DNA is a double helical structure resembling twisted ladder or a spiral staircase. You might have seen in the market. There are spiral staircases also. So imagine that to be a DNA. In, and in that spiral staircase, you might have always also noticed that when each step you take, it is rotated at a different angle. Okay, so imagine that to be a DNA. Within the double helix, I have already discussed about the base pairing. Anti-parallel strands are there. Spiral structure. Spiral structure. DNA is a right-handed spiral. Right-handed spiral. Okay. It means it will rotate in this way. And the DNA double helical structure is twisted in this way that it forms a major groove and a minor groove. And these grooves play a very important role in further molecular biology and protein DNA interactions. And it has been observed that Watson and Crick's model was completely consistent with X-ray crystallography given by Franklin and Wilkins, which showed that DNA had a regular repeating structure. So now we'll be discussing about the double helix, which is existing in multiple conformations of DNA. So here we have A, B and Z DNA, and the basic is, you can see that A is much shorter and broad, while B is longer and thin, and A is even longer and thin than Z DNA. So you can take of it as like motu, medium, lambu. Okay. And the here are the basic things that, but one which you can just go through. I won't be explaining it because then you'll get bored. But one thing I will tell you is that although ADNA is much shorter and broad as compared to BDNA, but it has more base pairs in per turn of helix. Why? Because the rise per base pair is less than that of B and Z DNA. Okay. The DNA helix rotation, that is in this way, helix rotation means it is right handed. This I am telling, this arrow you can see that it is in right, this arrow, when you will match it with your hand, so A and B DNA will be, you will be matching it with your right hand, and Z DNA will be matching with your left hand. And groove proportion you will see is that major groove of A DNA is extremely narrow but deep, while B DNA is of wide and intermediate depth, while Z DNA is flattened out on helix. You see, flattened out on helix. Minor groove proportion is of A DNA is very broad but shallow. This is minor groove. While for B DNA it's of intermediate depth and narrow, while Z DNA it is of extremely extremely deep and extremely narrow. Glycosyl bond formation means that bond conformation means it is either anti or in syn fashion. For A and B DNA, it's in anti-fashion. Anti-fashion means the base pairs will project away from the ribose sugar. But in case of Z DNA, for cytosine, it is anti-configuration, but at uh, guanosine, when it is the case of guanosine, then it becomes syn conformation. And helix packing diameter means the diameter of this helix. So it is the least for ZDNA, that is why it is elongated and slim, and more for ADNA, and that is why it is shorter and broad. While rotation per base pair means that it has a, between each base pair there is an angle of 36 degree, and that is why there is a turn. Got it? Are other structures also, that is a pDNA, which is deduced from molecular modeling, and it is 75% longer than bDNA with just 2.62 bases per turn, and it consists of mainly guanine and cytosine. And here you can see there is a triple helix also, which we call it, and the base pairing triple helix. Why? Because see, there is triple bonding, TAT, CGC, like that bonding is going on, and we call this Hoogsteen base pairs. But in detailed discussion is out of the scope of this video. As I already told that DNA spiral structure is due to its base pairs, and it is this spiral structure which conforms to its stability. So as we increase the temperature or increase the UV radiation effect, it will cause the DNA to dissociate, because these are the extreme conditions. But even if you try to heat it and stop it, you try to uh, start cooling, it will again start to renature. And DNA quantification methods is first based on absorbance, that is your UV, normal UV absorption spectroscopy, that is it will absorb UV radiation, and then based on that absorption concentration is being calculated. And dish test is based on the reaction of diphenylamine with the sugar of DNA, which forms a blue complex, and it is further subjected to UV absorbance, and then that's how its concentration is being calculated. Methods based on fluorescence. The fluorescence, it calculate if the concentration of DNA is calculated on the basis of the fluorescence it emits, and it is mainly specific to dsDNA and RNA, and it is of much high sensitivity, that is it can detect even small quantities. So I have mentioned here A260 by 280, A260 by 230 ratio. So it is this ratio that we use to quantify our DNA and RNA. So 260 by 280 is for RNA, while 260 by 230, one sec, just one sec. It's the secondary measure of purity for DNA or RNA. And there is other methods based on electrophoresis. You might have heard about gel electrophoresis. This gel electrophoresis, what we do is we prepare DNA samples, and it is uh, we store it in sorry, the DNA samples are being prepared, and it is added in equal volumes onto the gel, and a gel is being run. So wider the band, and in the end, after the gel run is being completed, there are various size of bands like this which will come onto the gel. So wider the band, more it has DNA in it. And it is this gel electrophoresis that is being uh, visualized using a trans illuminator or a gel documentation system. So that's all for today, and I hope it has given a basic understanding of the structure of DNA. And if you have any doubts, you are most welcome to post it on comment section. Thank you.